Okay, hello everyone. Welcome to the Great Lakes Seminar Series, co-hosted by the Cooperative Institute for Great Lakes Research and the NOAA Great Lakes Environmental Research Laboratory. My name is Greg Dick, and I'm the director of SIGLR, and it's my sincere pleasure to introduce today's speaker, Dr. Rebecca Klaper. Rebecca is the Vice Dean and a professor at the School of Freshwater Sciences at the University of Wisconsin-Milwaukee. Professor Klaper studies the potential impact of emerging contaminants like nanoparticles and pharmaceuticals on aquatic life and how we may design these chemicals to have the least environmental impact. In addition, she has examined the transport of these chemicals through the wastewater treatment systems and into the environment and how different treatment technologies may remove them from the waste stream. Her research links the impact of these chemicals on the health of aquatic species to that of human health. She uses genomic technologies to determine how these chemical stressors may impact organisms and how the biochemical response may dictate sensitivity or resistance to stressors. As director of the Great Lakes Genomics Center, she seeks to support other researchers that are using genomic technologies for environmental research. I also just want to note that I'm very grateful and thankful to have Rebecca on our SIGLR Council of Fellows. Thank you for doing that. Before I turn it over to the speaker, I'd like to remind everyone to please leave your microphones muted for the duration of the webinar, and you're welcome to enter questions into the chat box at any time during the presentation, and we'll moderate that at the end of the presentation. So with that, I'll turn it over to Dr. Rebecca Klaper. Welcome, Rebecca.
Thank you so much. Thank you so much for having me for this talk. I look forward to sometime in the future when I can meet folks in person. But thankful for this technology to do this way right now. So as mentioned, I came to the School of Freshwater Sciences, which was back then was the Great Lakes Water Institute back in 2003. And I began my research on emerging contaminants, so pharmaceuticals and personal care products that were entering the waste stream and into sewage treatment plants from people using them and then entering the environment from there. And I became interested in this topic as a grad student, actually. I wasn't researching this, but there was a great paper by the U.S. Geological Survey, Dana Culp and other folks that were looking at the presence of these compounds in the freshwater environment. It was the first time I had thought about how everything we use has a potential of ending up out in freshwater systems. And so the first thing I did was start out with traditional toxicological assays, but began building in genomic assays as a way to study how chemicals were having impacts on freshwater organisms, and did they really mimic what happened in humans that were using those compounds. So one of the questions that came to me as I started my job, and people in the Department of Natural Resources knew that I was studying pharmaceuticals, they were hearing about how birth control, they had read the same articles I did and heard about birth control pill medications going out in the environment. And they were also seeing changes in perch populations in Lake Michigan. And those of you who are from the Wisconsin or from maybe other parts of the Midwest, perch is a major, huge deal in Wisconsin for our fish fries, which have gone from being just a Catholic Lenten exercise to eating fish on Fridays to main staple of Friday food items in restaurants across the rest of the year, whether you're in a Chinese restaurant or a German restaurant or whatever. And perch are one of the favorites for these fish fries. And so the population crashing has been a big deal, and they've been going down since the mid 90s. And there are some indications this is due to zebra mussels taking out the bottom of the food web, and potentially to the fishing nets that were being used for catching perch. However, what the DNR folks came to me talking about was the fact that they were seeing changes in the gonads of these fish. And so when they collected males, normally you would see up on the top right here, white milky gonads, especially when they were in reproductive stages. And what they were finding is that all the males look like this. They down at the bottom, these kind of atrophied, weird, yellowy looking gonads. And so that's not going to be caused by a fishing regime necessarily, or potentially could be a food issue, but they were curious because of them hearing about the potential for pharmaceuticals going out in the environment. Was it, birth control pill medications? Maybe we're changing the gonads of these fish. And so it became another focus of what I was doing. Could we tie any of the chemicals that were coming out of that sewage treatment plant to the health of these perch. And I'll say the perch, the adult perch, sit at the outfall of the sewage treatment plant. It's nice and warm. There's lots of food there, little critters, and so it's kind of a bad design for exposure. They're really sitting at the place where they're going to be most heavily exposed to this stuff. But the question is, what chemicals could be causing an impact if it is a chemical cause? When you look at what is used in the population, there are over 350,000 chemicals and mixtures registered for production and use, according to a recent study in environmental science technology. This is 3 times the number that was previously estimated to be out there. And we don't really know what the identities are for some of these compounds. And so the fact that we don't have information on even what these chemicals are means we have less information on what impacts they might cause in the environment. And then how do you narrow down which chemical or which mixture of chemicals out of the suite could be causing a problem? It really becomes a kind of a seemingly insurmountable problem. There are also limited numbers of tests on the chemicals that we do know about. There's a lot of chemicals and only some of them are in the databases. There's also little data on much more than acute toxicity for a lot of these compounds, and definitely less on organisms that are not, or tissues that are not human related. So limited number of species have been studied. The time scale is off, so a lot of species are being exposed to small concentrations over long periods of time that may cause impacts on growth and development that might not be measured in these tests. The model organisms may not be appropriate. And so there are lots of issues, the fact that we just don't have a lot of data. At the same time, we know that emerging contaminants, anyway, are found in freshwater systems across the United States and in areas where we see impacted aquatic populations. And so it's this storm of events where we have not enough information, and yet we know that there could be a potential for these compounds to have some kind of impact. However, when you look at the reality of the situation, there are lots of things that could be affecting organism populations, from single chemicals to mixtures or other stressors that are out there in the environment, whether it's climate change, to things that are going on in our agricultural systems, to invasive species, to droughts, other things. So how do you separate out this soup of stressors and what might be causing the biggest impact, or the top 5 things that you'd want to tackle in order to protect organisms in the environment? So there is some hope. Molecular tools have come online and really have exploded in the last, I'd say, 10 years. When I was first a graduate student, when I was an undergrad, the Human Genome Project was just getting majorly underway and was finishing when I was a grad student. And really the focus was really on humans and trying to figure out our whole genome. But since then, we now have so many tools for dealing with other organisms, many other genomes sequenced, lots of tools that can be translated going across organisms. And so there's been an explosion in what we can use these tools for. So it's not just human health now, we can potentially use them for environmental health. So one of the things that the EPA has been invested in is looking at how chemicals could be linked to changes in pathways within organisms, and then networks of genes that might be impacted, or pathways, how that influences the state of the cell and the health of cells and tissues, and then up to things that might be relevant for risk assessment. So, the growth, the reproduction, and the survival of organisms. And it may be a way to provide a mechanism to separate some of the impacts of some of these compounds if only certain pathways are turned on with certain chemicals. It might be an answer to what's causing the biggest impact too, if you can get enough of this data linking the chemical structure to some of the molecular things, to some phenotypic impacts that are happening. So here's where our research started. We looked at what's being found in the Great Lakes. And so when I first started at the School of Freshwater Sciences, there wasn't a ton of information on the Great Lakes or huge bodies of water. And really the adage was that, the solution to pollution is dilution. If you look at a huge water body, you're probably not going to find a lot. And so if I was really going to get any kind of realistic idea of what organisms might be exposed to, I had to go out and do a study. There were some studies on tributaries and maybe on one of the other lakes, but really if we're talking locally perch populations, we set out to get some more information. And so this South Shore Water Reclamation Facility, sewage treatment plant, is sitting just south of Milwaukee, and it takes about half of the sewage from Milwaukee, and there's another site just to the north Jones Island, that takes the remainder. And so we did a separate study, which isn't being shown here, where we looked at what was going through the sewage treatment plant over a 24-hour period, looking hourly at the changes that were going on in the sewage treatment plant. And then over the next 24 hours, we go out and sample in Lake Michigan. And so this was the companion study to that. And so we would match what was going on in the treatment facility to what we found out in the lake. And so some of the things that we found were what you would have expected at that time, given what we knew about sewage. Excuse me. And that caffeine was one of the major tracers that people would use for looking at sewage leaking out into the environment, being emitted from wastewater treatment plants. And so we did find some of that. Each of these boxes is a 25 nanogram per liter measurement. And I believe, if I remember correctly, the blue is actually caffeine. But what was really interesting is that we were finding many other types of compounds too, even going out several kilometers to the east and to the south, which are the dominant water movement patterns in the lake. From this point anyway. And the other thing that was interesting is that we found these yellow boxes was a drug called Metformin, which is a type 2 diabetes medication. I had not heard of at that time, but it seems like there's quite a few people in the population that are taking this medication, especially in urban populations. And we found a lot of it going out into Lake Michigan. And so I figured that might be a place to start, but the question is that really something that could cause an impact to reproductive systems in perch? So we looked at the total average discharge out in the Lake Michigan. Metformin was actually by far the top thing that we found on a daily basis and then over the year. And so we estimated, based on our average concentrations, that there was 14,000 pounds of this going out from that one sewage treatment plant per year. And caffeine was probably the next highest one. And then a smattering of other things, acetaminophen, pain reliever, naproxen, also anti-inflammatory pain reliever, some antibiotics, as well as triclosan, antiseptic, ampicillin antibiotic, diltiazem, which is more neurologically related. And so there were a bunch of things that could be potential have potential impacts on organisms that we wanted to study. And so we also went to the toxicology databases and looked to see what kind of impacts they had. And my student Ben Blair created a risk quotient for each one of these compounds that we found out in from the sewage treatment plant. And what we found is, if you have a risk quotient of 0.1, it's of concern, and then of course higher is of greater concern. The dark blue lines are right at the outfall of the sewage treatment plant, and then as you go to the right, it's farther and farther away from the sewage treatment plant. So you see the highest impact, of course, in the sewage treatment plant. But what you notice is that for a lot of these medications, they reach above 0.1, even farther away from the sewage treatment plant, which was concerning. The other thing I'll point out is that there are only a limited number of compounds listed here. We measured like 60 compounds. And so sometimes it was because there really wasn't very much of an impact as seen in the literature. But also, in an equal amount, there wasn't enough data to actually create some kind of risk quotient for these compounds at the time. And so we're really missing, especially in 2013, we're missing a lot of information on quite a few of these compounds and what they could do in the environment. So we started studying things chemical by chemical. And as I said, metformin was the one that was coming out to the greatest extent in the environment. And this is a paper that we did it back in 2015. We did some studies on perch. Perch are very hard to deal with in the lab, and we found some indication of endocrine disruption. That hasn't been published, but we looked at vitellogenin expression, which is a bioindicator of endocrine exposure, endocrine disruption, exposure to endocrine disrupting chemicals. And we did find some vitellogen. So we moved to fathead minnows and looked to see if they're native species, of course. They're widely used in toxicology in the United States. And they're freshwater organisms, and they are dimorphic, where the males end up with this dark fat pad and they have darkened body coloration, and the females are much smaller, and often, if they're pregnant, then you can see the eggs developing here. And what we found is a significant decrease in the metformin treated organisms, particularly in the males, in their size and their growth. Over time. And then also the other thing that we found is that there was egg development within the testes of the males that were exposed to 40 micrograms per liter of metformin, which was comparable to what we were finding coming out of the sewage treatment plant on the heavier days. And we also found that there was also a decrease in reproduction. And so we did some more focus work on this particular chemical, but on a chemical by chemical basis, this was an indicator that at least one of the medications coming out of the sewage treatment plant may be partially the cause of what we were seeing in the perch. One of the things that, so when we started doing that research, we didn't start looking at reproduction right away. We're looking at those physiological impacts because it's, metformin is not meant to be as something that acts on the endocrine system. It's a metabolic drug to impact, insulin regulation within vertebrates. And it doesn't really look like an endocrine disruptor. It doesn't have those, I don't have an example here, but the circular structures that the hormones have. And it's not predicted to bind to the estrogen receptor, according to the molecular assays. But what it is a drug that's being prescribed for polycystic ovarian syndrome, where it lowers progesterone levels in females. And so polycystic ovarian syndrome, you end up, a human ends up developing multiple follicles at the same time, ends up leading to problems with fertility and reproduction. And I think it was an accident where someone who was diabetic was being prescribed the metformin drug, and it also ended up leading to fertility in that case, and they started being a case study, and now it's being prescribed, I think, more widely for PCOS in women, which means that it's affecting their reproductive system somehow, and again, then this made it a greater candidate for something that might push endocrine pathways within fish. And so we started looking, and at the endpoints, it does affect this last box down here in the liver. We did find changes in PXR and estrogen receptor and phytalogenin and CYP3A4, which is towards the end of this pathway. What we've been doing since then is looking farther up the pathway to see if we see changes in GnRH. Another colleague of mine has found changes that happen up in the brain farther up as a relation to the exposure to this particular chemical. So it is potentially an endocrine disrupting chemical. And again, we found changes in phytalogenin that relate to this as well. And the gene expression also relates to that change in histology that we saw in the gonads. And so these pathways most likely have some kind of relationship to the histology that we saw, where we saw those intersex fish. So that's just one chemical, though, and as you saw from that chart, there's a whole suite of chemicals that are coming out of the sewage treatment plant. And so the bigger question is, how do you separate out all those other effects that might be happening? We know from some of our other work and the work of others, of course, that you can have single chemicals where it doesn't necessarily look like there's a change. This is an example of lineuron, a pesticide, a DHP, a plasticizer, and which alone at environmentally relevant concentrations don't necessarily affect testosterone in fish. But if you mix them together, it has a significant impact on the testosterone production in fish. And so mixtures could have more than additive impact if they act on either related pathways or similarly affected pathways. And so it's a real problem in trying to estimate what those mixtures might be doing. So one of the things that we did, and this was a former postdoc of mine, was look at some of the locations that related to where we had done those chemical collections, and took the water and exposed zebrafish embryos and looked to see if we saw some of those pathways in those mixture exposures, basically. And so one of the things that we found is that the places where we found the highest level of compounds, we were finding things like higher up in that pathway, like that GnRH in the brain of the organism, and as well as down at the end parts of that pathway, estrogen receptor, when they're exposed to mixtures of compounds all over the system. But the other thing to note is that there were some places where there wasn't really any effect even from that mixture. And so it could be that it really is just certain chemicals that are higher in certain places that cause that impact, and trying to separate those things out can be very difficult. This is a study that was just published in Environmental Science Technology, not by us, but by U.S. Geological Survey, where they were looking in West Virginia at a watershed, and they tried to do the same kind of thing where they're estimating out of all the chemicals that could be in that watershed, which ones are causing the impact. And you can see there's quite a few when you look individually that in looking at acute PNEX, that 72% of the compounds have some kind of indication of risk, where that risk quotient is less than 0.1. However, when you aggregate these into a risk index of all the chemicals together, most of their stream reaches pose some kind of risk to aquatic organisms. So the mixtures of these things together could end up having a huge impact. And it's difficult to separate the impacts of individual chemicals. This is back to some of our research. It hasn't been published yet, but this was work with, and I mention it because it's work with NOAA, since I'm talking within the Siegler group, where we were working with NOAA Mussel Watch, and they were placing zebra mussels around the Great Lakes and looking at chemical accumulation in those zebra mussels. And at the same time, we took samples of the gills of those zebra mussels and looked at gene expression in relationship to the chemicals. Some of the things that we found is that it's very difficult to separate those chemicals because there's a lot of overlap, and the increase of one chemical ends up leading to the increase in another. However, we do find that there is some correlation of particular genes that are related to stress, aryl hydrocarbon receptor, and others that are related to particular chemicals. So here, this is the sum of PCBs or sum of pHs, but we also found that not only the legacy contaminants, but some of the emerging contaminants could also relate to those particular compounds, and they weren't necessarily found in the same patterns as the legacy contaminants. So things like triclocarban and 4-nonylphenol were also associated with certain genes, sometimes different ones like this heat shock protein. And so it can be a challenge to separate the impacts of individual chemicals, or which ones need to be cleaned up, for example, because of their co-occurrence in these systems. If you're really going to focus on a cleanup event in an AOC, which chemical should you be focusing on to be most effective? So the other question is, how do you evaluate large number of chemicals? I mentioned at the beginning that there's huge numbers of chemicals that we just don't have a lot of information. And so there's been a move to start using high throughput data and molecular data to complement some of the environmental sampling that's gone on. This is another example of some of the US Geological work, survey work, where they have looked at water samples, looked at all the analytes that are in the water samples, and then exposed those water samples to cell cultures or other high throughput screening tools that are molecular to look at where they're seeing bioeffects in the sum total in order to identify impacted watersheds. And then on the flip side, also using that data to say which chemicals might be important. But there's a problem. There's not often solid links between in vitro studies and molecular studies, these gene expression studies, and in vivo responses. And so this is where folks are trying to build up adverse outcome pathways. So where you look at some of the initial molecular information and see if there can be a clear tie to something downstream that is something that we would normally use in a risk assessment. So there can be some of the reasons why we don't necessarily have equivalence between in vitro effects and in vivo responses is that it can be there can be changes in uptake or distribution. It's not necessarily even. So looking at a cell culture versus a whole organism, you might not see that it only goes to one place in the organism where it doesn't have an impact, or it really does have an impact. The molecular changes that you see can be adaptive. So one of the common things that folks measure is oxidative stress. And oxidative stress is an adaptive response and a protective response. Even the heat shock proteins, protein folding assays, they're protective response to the organism. And so it could mean that they have turned on and that the organism doesn't show any kind of response because the protection mechanisms are working. We also don't have any information on enough pathways. This is really where we're building data right now to have the most powerful tools. And again, providing that link to apical endpoints that we care about. So right now, it's thought that it's definitely a great screening tool, but that we need to move beyond the models and the in vitro tests to make these stronger links in order to use these as a definitive tool for risk assessment. So one of the things that we've done more recently, and this was with Dana Kolpin and Greg Lefebvre in Iowa, is looking at how we can link the chemistry, some of the biological effects in the real world. And can we see a clear link between some of the contaminants of emerging concern and some of the impacts that we would see both molecular and also phenotypically within organisms. So we've just started with the molecular work. We've been building in some of the phenotypic work most recently. I don't have data on this in this presentation, but stay tuned. So one of the things we were looking at is the stream that's very heavily wastewater impacted in Iowa, and looking at some of the molecular pathways that are turned on in relationship to these changes in many chemicals. So these include pesticides, as well as a lot of pharmaceuticals and personal care products within these streams, and using an upstream versus a downstream kind of model. So upstream and sewage treatment plant versus 2 places downstream, and looking at how these things vary over space and time. So samples were taken over every month of the year. And then also we built in an in-stream test where we put fish in the stream. And some of the things that one of the major takeaways that we found is that there's huge variability in the stream, but also there are a lot of different pathways that are turned on in relationship to that exposure. One of the things that we've been trying to do is do similar to what the US Geological Survey has worked in, but look at some of those molecular predictions from ToxCast, for example. So when we measure any of those chemicals, we would then take this to the ToxCast databases, where they have looked at some of those in vitro screening tools, and look to see, based on the concentration that you'd see in the stream, where would you anticipate to see an impact, and on which kind of pathways in the organisms. And so some of the things that we find is that there are particular pathways like aryl hydrocarbon receptor and HRH1, where we end up seeing impacts going from the effluent, going all the way across all of our time periods. And however, that there are some pathways that are missing. When we do more gene expression assays, there are some places where we're missing information. For instance, in the histaminergic system and cardiovascular system, we found a lot of indication in our genomic assays that those might be potential impacts, and yet there's not necessarily great tools for looking at. Thyroid is another one where they're still developing tools that are useful for looking at impacts of the thyroid system. So here we turn to things like next generation sequencing, and the benefits of this. You take a sample and you basically sequence all of the RNA in the cDNA version of the RNA through next generation sequencing tools. And the benefit of this is you have unbiased measurements of the processes that are going on in cells. There's no predetermined pathways for your test. You can really probe and do discovery on what other things might be impacted. You can look at different organs and tissues and provide a really detailed analysis of the pathways impacted. The drawbacks of this is that you're looking at a snapshot of time, and so the mechanisms could change over time as the organism is first exposed and then is processing and adapting to the change in exposure conditions. It's not necessarily tied to an adverse outcome, just like the other in vitro assays aren't. And there's huge quantities of what can be very noisy data. And so this is a huge thing that we're always struggling with, is the amount of data that's produced and whether it's a true signal of what we're seeing in the organism. And there's also not enough genomic information for organisms of interest and pathway information of organisms of interest. Even though we can make we've made huge strides in linking genomic pathways going across many organisms. A lot of the ones that we really care about can be very different evolutionarily, and there are a lot of unannotated species, so where we haven't really identified what the genes are or the pathways in particular organisms. Fish are a particularly interesting example because of the ploidy level in the fish can be really crazy when you're trying to annotate a genome. And so we're missing genomic information that I think once it comes along will provide us a new way of analyzing our data. However, what we have found is that there are some novel pathways that are instigated by these exposures in these environmental conditions. So what we find, there's definitely differences in the upstream versus the downstream in regulation of many genes. And that there are certain genes that are impacted that are not commonly used for monitoring for exposure to sewage effluent. This includes adrenergic signaling, some cardiac signaling for development and also muscle contraction, neurological pathways, immunological pathways. And so what we're doing is identifying different biomarkers that you might use for exposure to wastewater, and that also could be probed for the potential impacts they might be having on species that go beyond our traditional toxicological measurements. Lastly, one of the things that we're doing in our lab too is looking at how those molecular changes end up relating to sensitivity of different species. So a lot of the assays depend on things like Daphnia or zebrafish, maybe flies, chironomids, but there's huge differences in sensitivity when you go across these different species, and there isn't a real clear indication as to why. It could be that they are exposed to a different degree. Daphnia take up a lot of stuff from the water column. Chironomids are found in the sediments. It could be that there is true sensitivity. We know some fish species are very sensitive biochemically to particular toxins. Here's just an example. One of the other parts of my lab is looking at things like nanomaterials, manufactured nanomaterials, and trying to figure out what kind of impact different types of nanomaterials might have on organisms. And so here's just an example of nickel manganese cobalt oxide, a particular type of nanomaterial that's used in solar energy and battery materials that has a potential for leaking out in the environment. And you find differences from going from a Daphnia, which is very sensitive, to a fruit fly where we really don't see any impact. So why is that? So one of the things that we found, we did a huge exposure where we looked at several different concentrations, a couple different time points, looking at chironomids versus zebrafish versus daphnid exposure to these compounds, and seeing what kind of things were happening in these organisms. Up on the top right here, what you can see is, for example, that in chironomids, we really just don't find a lot of impact of exposure at a 1 milligram per liter exposure level in the media. For chironomids, there's not a lot of things being turned on in these chironomids. However, in zebrafish, there's a huge amount of change going on from 1 to 10 milligram per liter to ions, an ion exposure, which is a control of the stuff leaching out of these nanomaterials. In daphnids, we see comparably fewer genes being expressed, but they're at a much higher level when you look at daphnids. One of the important things that we've noted, though, going across these, is that there are certain pathways that are lit up in each of the organisms. And one of the things that we found, actually thanks to the chironomids, which change color from this red color to clear when their heme proteins are dysregulated, is that these nanomaterials are greatly impacting the chironomid heme production, which relates to iron protein synthesis within these organisms, and which ends up leading to lower heme levels, disrupted growth and metabolism, because those heme proteins actually are related to a lot of different things about metabolism in these organisms, and all organisms actually, including plants, that is related to photosynthesis. So, and then we also see small size and delayed adult emergence, and it turns out that this is a common theme going across all of these different organisms. And we see genes related that are iron sulfur protein related genes going all the way through each of these organisms. And they're different ones, but they all kind of relate to the similar kinds of pathways, and they're disrupted to varying degrees. So there are some in chironomids, many more in daphnids, and different ones in zebrafish. So it's a way of linking up why organisms may be sensitive by finding common pathways going throughout them in order to ask new questions about sensitivity. So overall, these new tools in our toolbox hold promise to address some of the complexities that we see in environmental media, identifying maybe the most likely candidate for impacts in a mixture of exposures by looking at which pathways are turned on and which chemicals those might likely be related to, identifying environments that need greater monitoring or cleanup by using these molecular tools as opposed to some of the other, especially ones that are verified by some of the apical endpoints. Identifying which species are most sensitive and why are they most sensitive, and overall creating a more complete set of tools for predicting across important apical endpoints. So using the molecular information to provide us information about what's going on within the organisms, rather than just relying on a gross tool in order to figure that out. So with that, I'd like to thank a lot of our funding organizations. I've left out a couple of students here that are my more recent students, but this is funded through the last part was from the Center for Sustainable Nanotechnology through the National Science Foundation. But we've had funding from our local sewage district, the Argosy Foundation, Wisconsin Sea Grant, and others in order to do this work, as well as NOAA. And with that, I will say thank you very much, and I'd be happy to answer some questions.
Thanks so much, Rebecca. Hi, everyone. This is Nicole from the NOAA Great Lakes Environmental Research Lab. I will be monitoring the chat here as well as our attendees list. If you would like to raise your hand, I will attempt to unmute you, or you can put your question in the chat and I will read it out loud. But we look forward to having a conversation.
While those questions are being typed, I'll ask the first one if that's okay with you, Rebecca. This is Greg Dick again. So this question's a little bit out in left field. You mentioned some of the challenges with interpreting next generation sequence data, the massive data sets and complicated data sets. And of course, environmental microbiologists are starting to use these approaches or have been using these approaches directly in the field, right? And then of course, there's the whole eDNA crowd looking at genomic data, which just makes me wonder if there might be a day when we can use those techniques to, as surveillance directly of field populations. What are they feeling in the environment?
Yeah,
is that feasible?
That's definitely a hope, and I think we're getting closer to that. I think once we find, vitellogenin, for example, is already being used as an indicator of when organisms might be exposed to an endocrine disrupting compound. eDNA, for the presence of particular organisms over time in areas, notably around here, Asian carp. Have they been present? Is there any potential that they've been present in a waterway? And so I think that there is, I think it's already being used to some degree, at least for an initial screening indication of what's going on, either in an organism or in an ecosystem.
Okay, thanks.
We have a nice question from our colleague John Bratton here from Limnotech. Can you comment on the changes that treatment plant processes that can remove more contaminants prior to discharge, for example, pharma?
Yeah, so there are, if there are some places where things like nanofiltration are being used, which take out many more of the compounds of interest from sewage treatment plant effluent. And I don't think it's, widely used in the United States. There are definitely some places, especially where there's more direct link between the sewage effluent and uptake into drinking water systems. And definitely, I know in Europe, Paris, around Paris, for example, they're using it in their drinking water facility to remove contaminants. So that's one that holds some promise. Although you have, I think, a decrease in the amount that you can process through those systems. I know our sewage treatment plant has also been investigating whether letting secondary treatment systems go anoxic every once in a while would get rid of things like phosphorus even within their wastewater treatment systems. One of the things that we did, we worked with Veolia International and did a research project with them on a nanoparticle treatment, which it was a very tiny amount of nanoparticles in a huge vat, removed 70 to 80% of the compounds within a treatment system. So it's just the fact of, what environmental impact do they have and can you get them back out after you use them for removal of contaminants because they are little tiny particles. So there are possibilities out there. The other question though is, if you don't know what kind of impact the compounds are causing, how much funding do you want to put towards the removal process? So the fact that we don't have information about the biological impacts of our potential environmental impacts on a lot of these chemicals, it makes it very difficult to make the justification of other things. How much do you need to remove? Which treatment process is the best, most economically feasible for the amount of removal that needs to happen? So without that information, it's hard for, I think, cities to invest in the technology without that push.
As a bit of a follow-up for myself, is there any indication on how these are getting into the treatment? Is it by discharge through humid fluids, or is it by humans, dumping things into their toilets, for instance, to get rid of their pills?
Yeah, I think, there was a big emphasis even around here still on take-back events and taking compounds out of the environment because people are dumping them all down. And I think that is, a portion especially hospitals. There's been a huge change in the regulation of how things get disposed of in hospitals, and so they're not just dumping everything down the drain now. There are different buckets for disposing, especially of the regulated medications, the heavily regulated medications, where before they had to legally dump them down the toilet. And so hospitals are a big one that's been targeted for removal. As far as the general population, we did a study where we went and counted the pills that were being brought back, and we haven't published this data. I still have it sitting on my things to publish data set, but it was from a while ago. It was some of the first take-back events, and what we saw, the things that were being brought back largely mimicked what people took in the population. It was, I'd say, heavily biased though towards the neuropharmaceuticals. People would, I think, my hypothesis would be is that people take those for a little bit. They don't like the way they work, or they don't think they're working, and so then they're just sitting in their medicine cabinet. Some of the other things that were brought back, there would be buses that would pull up to give us stuff for take-back events that were from old folks' homes, because they had all sorts of samples and medications that were not used from people. And so there's some targets there for removing stuff from the environment, I think, that maybe folks don't think about. However, that being said, I think the consensus is that most of this is coming from people actually taking their medication. For Metformin, for example, there's huge quantities of the population that are taking that drug. And it's increasing because it's being prescribed now for pre-diabetes, which is a pseudo condition where you're giving some indication that you have some of the things that would lead you into a diabetic state, but there's no clear link between pre-diabetic and people that become diabetic, and yet they're being prescribed Metformin. It's being prescribed as a modern anti-aging compound, an antioxidant, and so I've seen where the advertisements for folks using that, and doctor discussion boards where they're prescribing this medication as an anti-aging medication. So anyway, so I think that a lot of it is people taking medications and they end up out in the environment because they go, a lot of it goes through you and then ends up in your excrement and then ends up in the wastewater treatment plant. And I'll say the wastewater treatment plants do a fairly good job of taking out stuff, and it ends up in the, the solids of the wastewater treatment plant, which then can get spread in the environment and also end up in water systems. But it just doesn't get rid of everything. And the more you have coming in for some of these medications, the more you have coming out.
Thank you for that. One more question related to pharmaceuticals specifically. Dr. Shima Katiba says, thanks for the presentation. I would like to know how I could measure the annual discharge of pharmaceuticals, if that's possible.
That's a good question. So our detection technologies have gotten better. However, it's still very labor intensive to measure each of these compounds. If you're measuring one, it might not be so bad, but it's a lot of people work going into determining the concentrations that are in each one of the samples. If you send it to a commercial lab, it can be $500 to $1,000 per sample. So it's really not feasible necessarily for a lot of places to do these measurements, which is part of the problem. And why it's not, widely monitored everywhere. It's mainly research scientists that are doing it. So, but we do this. There's some of the improvements that have been made. So when we first started doing our studies, we were doing extractions and then, injecting labeled standards and then running it on LC-GC mass spec. And but now there's some the U.S. Geological Survey, which is of course fantastic and leading the way on this, has done some direct injection studies. And so there's a move to going towards that where it would be less labor intensive for the processing of samples. Data analysis is still a bit labor intensive, but it might end up dropping the price for some of the analyses. So we're progressing in our chemical analyses as well.
Thank you for that. John Urquhart asks, as our understanding of pathways and potential hazards improves, do you imagine that we will screen potential drugs for impact to the environment prior to distribution to the general public, i.e. as a part of the R&D for drug manufacturing?
Yeah, that's a really good question. So right now, the Food and Drug Administration does not require that unless there's some indication that it could be an endocrine disrupting chemical, and then it's not required, it's suggested. And that was a push from the scientific community and the nonprofit community that they include some language. And it may be turned back that they ask for that information for approval of a drug. As far as any other mechanisms, though, unless you anticipate that the compound is going to be in the environment to a level that's greater than, I'm trying to remember the number, but there's a limit, and if you anticipate and through your modeling you determine that that chemical is not going out at a certain concentration, then you don't have to do any kind of environmental assessment. And so that's the problem, right? Some of the pharmaceuticals are active at a much lower level. Hormones were one, that's why they got kicked into that category. But the other problem is that if the drug, when it was initially projected for a particular population of a disease, let's say, and that was its approval, I don't think it's ever reevaluated again than if it starts being prescribed either off-label or for another purpose. So for example, Metformin was originally being prescribed for diabetes, but now it's being prescribed for about 20 other things. And so the levels in the population of pre-diabetes alone, there was an estimate that half the state of California could be on this medication if you included pre-diabetes and diabetes populations. And so if you have that many people taking this drug, obviously your estimate initially is way off as far as how much of that drug is going out in the environment. So we're missing something here about potential environmental impacts. And so there's a question as to, yeah, I mean, how do you push that forward in the FDA? I have no idea. Or is there a link, that needs to be made with EPA when it comes to some of these medications?
That's a good question. Thanks so much. So I'm going to go back up to the top here. Carson Brown says, thank you for your talk. Amazing seeing applied work like this. Have you included algal sampling and contaminant levels as a way of predicting contaminant load for species higher up in the food web?
Yeah, that's a really good question. I haven't, although we just started doing algal studies in our lab most recently, partly because of interest in one of my students. So maybe that would be something to come. Some of the compounds are active on algae, and that's one of the reasons that it triggers that safety level. So triclosan, triclocarban are antiseptic compounds that were used initially in hospitals as a wash for hands, but then a success of that in that product was they were put in everything. So for a while there, any antiseptic hand soap that you saw had triclosan or triclocarban in it, and it's toxic to algae in particular. So and now there's been such a push, mainly by the state of Minnesota. Their scientists there really looked at the contamination of that compound out in the environment, that emerging contaminant, and what it might mean. It also, by the way, once it hits UV and chlorination, ends up being a dioxin, so not great. And so algae does, form the basis of some of these risk quotients that we're seeing here. But as far as a mechanism for uptake into the environment, I personally haven't looked at that, but I would think that would be possible, especially if it gets stuck to the algae or in the algae.
Thank you for that. Kathy Lates has a question. Regarding the mixture studies that you performed on zebrafish embryos, the corian is a very effective barrier to the uptake of chemicals. Did you repeat those mixture exposures in other Z-fish life stages or other species? And did you see different responses and or sensitivities?
Hey, that's a good question. Yeah, so some of the things that we've done with some of the medications are looking at, because the developmental progress within a zebrafish is, it's amazing how many changes happen over the first 10 days in that organism. And so depending on which part of the life stage you look at will tell you, it differs in the gene expression that you're going to get for that organism. And we have a study right now looking at nanomaterials where we're doing that, and pharmaceuticals actually, where we're looking at different exposure time points within that developmental cycle. For the study that I showed you, we did 3 days and 5 days, and so after they come out of the corian, but we did not decorianate the organisms when we did that exposure.
There's a question from Marcia Silva. Marcia, I unmuted you, so you're live now. Marcia, unfortunately, I had to mute you again because all we could hear was wind. I apologize. If you want to put your question into the chat, we'll read that for you. Krista Grimm, I'm going to unmute you for your question now. You'll have to unmute yourself as well. Okay, we'll go back to the questions board then. Keep trying, keep raising your hand if you need to. Let's see, we've got James Beck with a good question. Are many of these chemicals water soluble?
Oh, that's a good question too. Some of them are and some of them are not. It kind of depends on the chemical that you're talking about. And so, but we still, and what we anticipated initially was that we would find a lot of the water soluble ones in the water column. And I didn't mention for that Great Lakes study, we looked at sediment as well. So we measured each of those compounds. I didn't show that data, but it's in the paper if you want to look at it. And what we ended up finding was we find water soluble compounds in the sediment, and we find water insoluble compounds are those that are not as soluble in the water column as well. And whether they're attached to sediment particles or just free floating, it's not totally clear. Yeah, it's interesting. It wasn't what we anticipated.
Thank you. We have lots of good questions in here, and I apologize if we can't get to all of them today, but what I will do is I will print these out and I will send them to Rebecca so that she can respond at her leisure. So let me get back into the questions. We've got, can you comment on where you choose, if not in-house, to analyze your suite of emerging contaminants and how you standardize this over the years with variable detection limits?
Yeah, that's a good question. Yeah, because the detection limits have changed over time. Yeah, so, oh, and if you're sending samples to different places, we generally, for one study, we'll stick with the same kind of procedure and the same lab for analysis. So our initial studies, we were working with the Wisconsin State Lab of Hygiene, and it was my grad student and I and Curtis Hedman over there that were extracting and running samples. It was, we were working all together as a team, either one extracting, one running the machine, and the other doing the analysis. And then now we're working with Greg Lefebvre over in at University of Iowa. And again, that study that I was talking about, that Iowa study, it's all in his lab. We have done some companion measurements. The U.S. Geological Survey, David Sedlak and other folks have analyzed those samples for different other kinds of compounds as well. And what Greg and USGS are doing are comparing their samples and their detection technologies. That's a separate study that Greg's lab has done, but it's a good question. And I think it's really important within one study anyway to stick with the same lab, same procedure, because you can have issues with detection, limits of detection, et cetera.
Thank you. We have another question here from James TG, sorry if I've pronounced your last name incorrectly. The problem of mixtures remains overwhelming to discern the particular causative combinations. Are there new strategies to resolve this?
That's a good question. I don't know. What we've tried to do is look at groups of chemicals that act on the same kind of pathways, and looking at those as a study, or then looking at using one of those compounds in a study where we're looking across pathways. So I think that molecular information might provide us some indication. If you look at things like the PCBs, for example, there's a standard based on just one PCB, and so I think if we can do that with other compounds or estrogens, use E2 as a standard, and everything is based back on that as to how effective it is in the same pathways. And so there's not a lot of examples of that, but I think that's one strategy that could be used for looking at those mixtures, basing everything on some kind of standard within a method of effect, and then starting to strategize mixtures based on that strategy of how impactful are they on that pathway, and then what happens when you stick those in a mixture. Is it a one-to-one, or is it really, if something is a quarter active here and a quarter active there, if you mix them together, does it end up being, half? How does it relate? So I don't know if there's huge strategies. The other strategy people are using those environmental samples. So saying, okay, well, screw it. We know there's a bunch of stuff in here, so let's look at where are the greatest impacts happening, and then can we target those areas for, in general, the pharmaceuticals, in general, the agricultural chemicals that we know are in the mixture. and hopefully that would decrease enough of them that it would cause some kind of decrease in impact of those chemicals on the environment.
Thank you for that. We have time for one, maybe two more questions here. So it looks like we'll have a good handful to a dozen left over. I apologize again. This is a good question, especially since you mentioned all these complexities such as you know, climate change, water level rising, drought, things like that. So Michael Haberfield asks, related, in the Metformin example, it looked like the HQs were sometimes only approximately 1 order of magnitude lower miles out in the lake compared to the wastewater outfall. Does this indicate that there are relatively high levels in the ambient water lake? Sorry, I have to scroll. Water across wide areas, I think is the last part of this. Sorry.
Yeah, the Metformin example was really bizarre. We expected it to really taper off and to be somewhat predictable. In fact, we work with some of the folks in our school that do modeling of particulate matter, the little tiny particles and isotopes that move around the lake to estimate water pattern movement. It was not what we expected at all. And so one of the things I think that was causing that is that it's a constant emission of these compounds out into the environment. And so, and some days it would be very high, and sometimes even in a 24-hour period, there'd be a period where it was super high for some reason, and then it would go down. And so I think it's just that variability and output, but the fact that it's constant, you end up seeing it going farther out. I will say that we took measurements up near our drinking water intake, which is farther north and is away from the water pattern movement, and we don't find it up there. So it's not that it's everywhere. It's really related to that effluent coming out into the lake. It's just that the movement and the breakdown is a little bit of a mystery. There's been some industry reports. So one of the things that I also find problematic with the Metformin example is that it was estimated to break down fairly rapidly in the environment, as are a lot of pharmaceuticals, and we just did not see that as the case, either in our lab experiments or from our indications of what was going on out in the environment from our environmental sampling. There's been an industry study since then that said, no, it really does, it breaks down, but there have been many more studies saying that no, it does not break down like what you're seeing. There's some studies from Europe, Alistair Boxall's lab, that find that the Metformin and their water systems going from the outfall to where they do an intake is an equal amount. It doesn't break down in the environment, or there's so much coming out that there's just no chance of it breaking down to any significant level by the time you measure it many, many miles downstream in their river system. So I think it's hanging out there for quite a while longer than we thought it would.
Thanks for that. The rest of these questions I will email over to you, but Marcia Silva did have her question, so we're gonna try this one more time. Marcia, I'm gonna unmute you, and you should be able to unmute yourself now. Doesn't seem to be working, Marcia. I apologize. Sorry about that. Well, thanks everyone. Thanks for your attentiveness, and thanks for these great questions. Again, I will email them out, and if you want to share this presentation with any of your colleagues, we'll have this up on the Sigler website within the next couple of days. And keep your eye out for more Sigler and GLERL Great Lakes seminars. We have them posted on the Sigler website. We have about one every month. And thanks once again, Rebecca. We really appreciate you joining us today, and we look forward to more collaborations in the future and hopefully getting to meet in person sometime in the near future as well.
Great. Thank you so much for having me, and thank you all for your interest, and I'd be happy to entertain questions through email or another conversation.
Great. Thanks so much, everyone. Everybody have a great day. Stay safe.
Bye.
Thanks, Rebecca. That was fantastic.
Great. Thanks so much.
Clearly,
Thanks for having me.
Clearly a lot of interest from the audience here.
That's great. Yeah, I hope it was understandable. So
Absolutely, yeah.